Hello and good evening. We are so excited. We have Ken joining us from Hydrospace, but before we get started, I have a couple housekeeping items that I'd like to share with everyone. So for Photo of the Month this past month, congratulations to ETFI3 for winning the Photo of the Month with the most votes. They get a $50 gift certificate provided by the club. And then, as you all are getting used to, because we've had some wonderful donations coming in, all the people who vote go into a random drawing, and congratulations go out to Texas_Reef, and they won a worldwide coral T-shirt that was donated by WWC. So very exciting, and congratulations. At this past Saturday's gathering and giveaways, there were there's one missing here from the notes, so I'll start with that one. Some of the members who put down their name on the slip, if you don't put your name on the slip, I don't know that you're a member, and you don't get announced, and you don't get something on your profile page from me. But a shout out to Cedar Hill, who won, donated by School of Fish, who was our host for gathering and giveaways. School of Fish donated a flipper. It's not written down on my notes, so bear with me. It's a flipper kickstart. It's both a kickstand and a filter for your aquarium. It's really awesome. And then Photon Blue, he won a Fluval waterproof mat. So congratulations, Photon Blue. And their baby was there, and his wife. So they had the little baby there. And then Donald US 2000. Steve won the Tropic Marin Lemon NP for nitrates and phosphate control. So congratulations to all of you as well. The upcoming events, as you know, the giant thing we are all looking forward to is the Frag Swap on March 9th. We're also including a potluck to get back to that hometown country feel, old school. Everybody's always saying, let's go back to the good old days. So we are doing that. And with that in mind, and Nick, thank you for staffing the club table, where we'll be doing silent auctions and the freebies. So if you have frags that you don't have someone wanting to buy them, you can always drop them off there at the club table as well. And we're looking for volunteers to help us with check-ins, helping people set up, passing out raffle tickets, and helping with the club table, both the free raffle, which is also open to the public. But everybody who comes gets a ticket. And then we also have to make sure the sellers stay stocked with tickets to give out to people as they buy things. And remember to bring your cooler, your money, frags to swap, and a dish to share. We also are looking for volunteers in May for Aquashella. That's May 18th and 19th. The club has a table. We're going to be along the back wall in the UV light section, the saltwater section. So stop by and say hi. But if you'd be interested in volunteering for the event, please reach out to me. There's a sign-up sheet on the website. And I have the abbreviated version of housekeeping here. The next thing I have written down is June 15th, which is the gathering and giveaways at at Aqua Studios. And I think that's up in the colonies with Brandon. So we're going back there. And as always, you'll see the latest information on the website. So check out events there. And this that concludes what I have here for housekeeping. So I'm so excited. I've been so waiting for this to come, and I've been researching. This evening, we are joined by Ken of Hydrospace, and he's going to be talking about the bacteria and how it's so important, as we all know. And so I'm going to turn it over to you, Ken. Thank you so much.
Hello, everybody. I usually start with questions or anything like that, but I could just start with my background a little bit. I've been in the hobby, I guess, since the mid 80s, and started a saltwater basically when we were doing under gravel filters and with power heads on top, stuff like that, with varying levels of success. And started working in pet stores in the early 90s, and really got into when I started selling corals in the mid 90s. And kind of that sort of solidified my interest in all this. So I kind of devoted my life professionally to this hobby and industry, and got my undergrad at the University of Oregon, and then moved on to do some post-grad stuff at the Aquarium Science Program in Newport, Oregon, which is a one-year program. Very intensive aquarium related stuff for people with a background in the biological sciences. So while I was at the University of Oregon, I got my first taste of live food cultivation. And that was actually for zebrafish, working in a zebrafish lab. And it's an enormous lab. Some of you may be aware, zebrafish are like a model organism now and widely used now, but that was the first place where zebrafish were developed for that purpose. They're kind of like the standard white rat now in the field of science, mainly because they have a really short reproductive cycle and they have a clear embryo, so you can look inside and see what's going on, which makes them really important to developmental biologists. But my work there was just to grow the fish. I was an aquaculturist. I had nothing to do with the neuroscience that they did there. And part of that was raising, well, almost all of it. My main job was cultivating live foods. So it was paramecia, which are a little ciliate, and we hatched an enormous amount of brine shrimp and some little things called vinegar eels. They're actually a little tiny worm. And I thought it would be boring, but it was actually pretty fun. The challenges were unique and always something different. You wouldn't expect that, but always changing, different contaminations, or we were studying how we could supplement things, or how we could augment the nutrition of these foods by feeding our foods certain foods. Anyway, so when I moved on to the Aquarium Science Program after I graduated, I got into phytoculture and was growing phytoplankton, where we were using it to feed oysters in a molluscan broodstock program, where they were just experimenting with oysters. And that was, I guess, for the first like massive commercial scale work it was that I did. These are like swimming pools full of phytoplankton, literally big swimming pools. You climb in and out of them with a ladder. So, but of course the process starts with a little vial and then you work it up into bigger and bigger. I found that fascinating. So I was kind of stuck in the live food thing, and that was kind of my deal, I guess, for like 18 years, something like that, before I discovered this stuff. Which is while I was working for Algae Barn, doing largely the same thing, cultivating phytoplankton and copepods, live foods, aquarium foods. And we were going to use these organisms in-house, the Purple Non-sulfur Bacteria, which I'll get to in a bit. But they were getting into macroalgae at that time and a whole lot of other things, and just kind of got distracted, and they moved further south of Denver in an area I didn't want to live in. So I stayed in the mountains in my cabin and developed this company. And I'm still learning. I wouldn't say I know everything about these bacteria by any means. They have been around since the 70s in aquaculture and wastewater treatment. So they're pretty well studied, even specifically for those applications. There's just a lot of papers to sort through, and even a lot of the most recent, some of the best research has come out in the last 10 years, really. So it's just really exploding, and it's kind of neat. There's no shortage of really interesting things that we're finding out about these bacteria right at a time when both probiotics and the microbiome generally are a kind of a new interest in our hobby. So I kind of got in at the right time, I think, with the right bacteria. And a little bit about that I can explain later when I describe what purple non-sulfur bacteria are, because though they're widely found in nature, including in the environments they're trying to replicate, nobody in the aquarium industry has really done much with these organisms, despite the fact that they've been used in aquaculture so much since the 70s. So mostly fish farms and very much on shrimp farms as well. But that there's a lot these bacteria can do, and if I try to list it all, sometimes I fear it'll sound like I'm peddling a snake oil, because it really does sound like they do everything. But they're incredibly ancient organisms. They're photosynthetic. They are among the first organisms on Earth, but also even the first photosynthetic organisms, cyanobacteria, and therefore all chlorophytes, and therefore all land plants are descended from these particular organisms. And so everything kind of evolved around them, including corals, certainly fish, other animals. So we have these natural associations with these bacteria that that emerged, throughout our entire evolutionary history, because they've always been here with us. And it can be pretty complicated and intricate and varied depending on the organism. They're used for everything in horticulture with plants to, like I said, in fisheries, even for various things there, bioremediation to improve water quality, and also to help the fish or shrimp digest their food better. They call it feed conversion in those circles. So it improves feed conversion, and as a consequence of that, they produce the host animal actually produces less waste. So they basically digest more of the food they eat and produce less waste. So they act similar to a lot of other probiotics, also in that they inhibit pathogens. So very wide ranging. They're kind of a do-all sort of bacteria. And the best part of all is they don't really have any side effects that are harmful. So as far as we know, there are no undesirable things that come out of using them. And they have been used a long time in big commercial applications. So I think if anything, if there was anything harmful, we'd have found out about that by now. We're also looking at we, meaning the scientific community, is looking at these bacteria in the field now and studying their interactions in various natural environments, including coral reefs. They form natural associations with corals, which is kind of cool. So at Algae-Born, when we were interested in these, we were initially just interested in feeding them to the copepods to improve the water quality, which was really bad. Anytime when you're trying to grow something at a high density, when you're looking at it from like as a business and you're trying to produce a lot of yield, in a given amount of floor space, you're always pushing the limits, and then the water quality goes south. So this was kind of something we thought would improve water quality. And then as these organisms consumed all the wastes, they would become a food source for the copepods. And we thought that would be particularly beneficial for the Tigriopus, the big red ones, the tiger pods, as some people sell, Reef Nutrition sells them as. They're the color is actually really, really important, because those are carotenoids, which are incredibly important for any animal's health. So that just became a nut, like just kept having layers of icing on the cake as I started to research these bacteria more and more, and started to find that there's so many uses. There was no way that we could. I was shocked, to be honest, that we have ignored them in our industry for so long, given that they have so many uses, and that they actually are naturally occurring, unlike so many of the bacteria that are bacteria in a bottle, as people that are available in our hobby right now. These are actually naturally occurring microbes.
Well, and beneficial too. I first stumbled across your product, like, I can't be the only one, but I will admit I overdosed carbon vodka trying to improve water quality, and then my fish are struggling to breathe, and I'm thinking, I've got nothing useful in there. So the bacteria that is like, it's got to be like god awful.
Mm.
Where's the good stuff? And you are the good stuff. And so I'm so excited that you're here to talk about your products this evening, because bacteria is everywhere. It's in us, it's around us, and um, the without the good stuff, the bad stuff takes over. Opportunist.
Hmm. Well, even that's a great example of that when we're carbon dosing. So when you're trying to export nutrients, for example, you have two options. You can either add bacteria to do that, or you can promote the growth of native bacteria by uh, adding some sort of food source. And the difference between the two is that if you add the bacteria directly, you are adding only those beneficial bacteria. Whereas if you're carbon dosing, you could potentially be promoting the growth of less desirable microbes, including Vibrio, things that are, pathogenic to corals. And in so doing, you're mainly feeding aerobic bacteria. So as you experience, I've seen this a lot in the stores I've worked in. People will overdose things like no pox, or however you want to pronounce that, and the fish are gasping at the top. And so this is a safer way to do that. It doesn't. These are anaerobic bacteria, by the way. So they will not. They're not. Their respiration doesn't involve the consumption of primarily anaerobic, so they don't consume a lot of oxygen. And that, then there are the other, implications with pH and so forth. When you carbon dose, all that CO2 that their aerobes are going to produce is going to be carbonic acid, which reduces your pH. So it's just it's a less sloppy way of water quality management, and kind of like what they find in the shrimp farms is that there's like a dual purpose there. It's like a food that cleans the water instead of polluting it, right? So the bacteria themselves, and we can get into this, are extremely nutritious, extremely nutritious. In addition to just acting as probiotics, but then by being in the animal's gut, the animal produces less waste, and whatever waste is produced is readily consumed by these bacteria. So it's just kind of a win-win-win, particularly when you're using them as a probiotic. So that would involve putting it in their food. That could be coral food, fish food. Could be administered any way like that. And that type of use has been pretty well studied too. If you go on and do like just a search for like, say, one of the species we saw, Rhodopseudomonas aquaculture probiotic on Google, something simple like that. I mean, you could spend weeks and I know reading all the papers that have detailed research, research that's already been done in that area. So, but yeah, that as you pointed out, that's one of the important things if you're trying to use these for water quality management. We like to think of these bacteria as a live food primarily. That's where my initial interest was in them. And as I said, all these other things, denitrification, they're capable of. That they're heterotrophs and they consume organic matter, organic wastes. All those are just different layers of icing on top of that cake, which is this incredibly natural, nutritious live food. So just real quick on that one. So the reason they're nutritious is they're mainly loaded with protein. So they're always at least 50% protein, but depending on the environment, they can be upwards of 70% protein.
Wow.
And unlike phytoplankton, they're not an algae, so they don't have a cell wall. So they're actually they don't have that cellulose, the fiber, if you will. So they're much more they're much more digestible and loaded with certain fatty acids and B vitamins. So especially stearic acid, oleic acid, things that normally are not. From what I've read, this is not an area I know a lot about, but prepared aquarium foods typically are lacking in like oleic acid in particular. So just really complementary. One more thing I'd add, I guess, relating them to phytoplankton, there are studies out there that show that they're best used together. There is no one food that's perfect. So, and these are no exception. There are a lot of essential fatty acids and lipids that they're not particularly rich in. But when you combine them with algae, you have that high protein content, which algae does not have, phytoplankton. And then you also have they're very rich in B vitamins, particularly vitamin B12, which is very, very important, often a limiting nutrient in marine environments. Something we don't talk about in our hobby much, but sometimes that's something that limits growth and we're not even aware of it. Vitamin B12 availability. So anyway, they used brine shrimp as a subject, as a experimental subject, but it could apply to any animal, really. We kind of have the same nutritional needs, and they found that using the two, neither one was better than the other. What was best was to use them in tandem. So it's kind of Algae Barn, as it happens, unsurprisingly sells my product now, and they have a lot of combo packs, which we're pretty excited about, because those come with their phytoplankton. It's 4 or 5 species in Ocean Magic. So it's using them together with ProBio, which is our Rhodopseudomonas palustris, is very nutritionally balanced. So again, a lot of people ask a lot of stuff about these bacteria because they have a lot of uses. But I get everything from, do they get rid of dinoflagellates, to can they kill? Do they stop tissue, RTN and all that? And there are potential pathways for those things to occur, but the main thing we want to focus on is their nutritional benefits. And a lot of those other things stem from poor water quality anyway. And nothing against reefroids, but if you've already got too much phosphate, for example, and dumping all this powder in the tank isn't really, that's already rich in phosphate, so we hear, isn't necessarily good for that situation. Here you have a live natural bacteria that has a natural symbiotic associations with corals, and instead of producing a lot of waste products, they actually consume them. So it's just multifaceted, but I think a lot of those other things, the denitrification, stuff like that, is exciting. But think about, if you produce less nitrate because your foods are producing less waste in the first place, you're addressing the problem at the source. With any live food, it's just these particular bacteria have other capabilities if they survive in the tank. And that's the if, right? So that they're just kind of bonuses. So we always focus on the food aspect and then kind of address those other things as little side benefits.
Those are huge side benefits. Absolutely huge. When you think about your water quality improving, and that gives the health to your corals and your fish, and what they're eating is so much more nutritious. And it didn't even occur to me. I do have pods and amphipods in my both my main tanks, but then I'm going back to culturing again. And I usually run my cultures not with phyto, but with spirulina. And I see this as being a great partner with what I've done in the past. And so I'm very excited about the yields I'll be getting with this, and didn't even think of adding it into there until this evening. So thank you. Yeah.
And if you're, any situation like that where you're using these to gut load, there are studies out there showing these bacteria being gut loaded into organisms like, so it was copepods. In that case, it was Apocyclops panamensis, and Artemia, and Moena, which is Daphnia, more a freshwater thing, but a cladacerin, so kind of like a brine shrimp or something like that, I guess. And what other? And then Brachionis rotifers. And in all cases, they showed that those organisms do eat these bacteria. They showed it in their guts. So the nutrition and even like the probiotic qualities of these bacteria were transferred directly to to the target animal that was being cultured. They've shown in hatcheries with sea bass, tilapia, all kinds of things like that. When you introduce them into the diet that way, lower in the food chain, and it just moves up like that. They have the benefits of the antibiotics these things produce. They inhibit Vibrio, which is, and that's well known and well proven. That is humongous in our industry because Vibrio is one of the top pathogens. Also, like I mentioned before, the carotenoids, which like astaxanthin and lycopene are incredibly valuable, especially if you are to purchase them and put them in a food. They're almost literally worth their weight in gold. Astaxanthin, it's ridiculous how expensive that stuff is. And this is a whole cell source, right? So I believe astaxanthin is fat soluble. So it has to be consumed with their food. You don't just sprinkle a powder on something and expect. It's better if it's eaten with in the whole organism. So in that way, it's actually more bioavailable to the animal that eats it. So with fish, again, we just recommend like soaking like their pellets in it for a few minutes before. Without binders, it's hard to say how much gets on the food, but any amount is better than none. And if you're going to put it in your tank anyway, the best place to put it is in the food. It'll either end up falling right off the food into the tank anyway, or it'll go right through the fishes and corals' guts back into the water as part of the waste that they produce. And well, that's even better. So
is this something that you would feed daily then? Because a lot of the bacteria, say like once every 2 weeks or something, right? But being that this is live food, this would be like a daily addition to the tank.
It would, yeah, it would be, or it could be. It's one of those things that you can't overdose.
Okay.
Yeah, it's never pathogenic, and it's not going because it's anaerobic. You're never going to have a bloom per se, like in the water column where it consumes all your oxygen. If anything, it actually decreases your BOD, your biological oxygen demand, meaning any organics in there that are capable of consuming oxygen. So it actually helps prevent anoxic events. Well, yeah, you would just put it in the fish food, they eat it, and it increases the feed conversion rate. Like I was saying, it basically just means that the fish digests more of the food it eats. That would include fiber. So these particular bacteria can degrade cellulose, which is pretty cool. Not a lot of organisms can do that, especially in the marine environment. In the terrestrial environment, that's mostly fungi that break down like woody materials, right? So the cellulose is the cell wall of plants and algae, and it's the most abundant substance on Earth because it's so poorly degradable. But these guys can break it down. That's why they use it in the pond industry to eat leaf litter, stuff like that. Gotcha. But in the animal's gut, say you have a tang or a rabbit fish, or a more herbivorous species of angelfish, and you put this on their nori, they're actually going to digest more of that food and produce less poop. Because these bacteria then use the cellulose as a carbon source. So while they're in the fish's gut, they're producing and releasing all these vitamins, and they're basically taking waste products and converting them into something useful that the fish can digest and add to its nutrition. And again, and just as importantly, I guess, they produce less waste then. And like tangs are like the goldfish of the marine world, right?
Yeah.
So it can be. So, and we've actually got feedback from people feeding tangs that said that their tangs poop they pooped less, but the poop even looked different.
Huh. Okay.
Yeah.
Awesome.
Yeah. I'm sorry, and I know I went off on a tangent there. I don't know if I completely answered your question, but
yeah, no, you did. Just curious if you're something that you'd feed daily. Versus
right.
Every other week or every 2 weeks, because a lot of those nitrifying bacteria bottles say like once a week, once every 2 weeks, something like this. But being that this is live bacteria, more used as a food base, you can't overdose it. Why not soak their food every day? So
right, yeah, that's a complaint we get too, actually. Is that well, if I have to use this every day, we'd say you don't have to, but you have accruing benefits if you use it every day, because they don't necessarily compete super well in an aquarium. Unfortunately, even though they're extremely abundant on a coral reef in a nitrogen limited environment, they in an aquarium where there's lots of nitrate available, other types of microbes predominate. So that's one reason, say if you're trying to seed the tank, you know, it wouldn't necessarily, unless you're really trying to, you had a bioreactor with an appropriate biomedium and stuff like that. And then you already maintained a low nutrient. That's one of the paradoxes with these. People say like, well, if I already have like no nitrates, why would I add a bacteria that, you know, why would I bioremediating the water? Well, there are other purposes, there are nutritional purposes mainly with the bacteria, but it's mainly that these particular bacteria, in addition to denitrification, they do the opposite, which is take nitrogen gas and turn it into ammonia. But they only do that in low nitrogen environments, like a coral reef. And without getting too deep into that, before I get outside your question again, that's one of the most, they figured this out in the 70s, actually. It's nitrogen fixing bacteria that allow coral reefs to exist in the first place. They called that Darwin's paradox. When Darwin first went to Tahiti, he was blown away by how the water was super clear, indicating that it was infertile. There was no phytoplankton, and yet they were so productive. And he couldn't figure it out, and no one did until the 70s, actually. There was a particular expedition there that noted that the nitrate coming off of the reef was actually the levels were higher coming off the reef than going into the reef. And then they discovered, well, yeah, there's nitrogen being biologically available nitrogen being produced on the reef by these nitrogen fixing bacteria, which, by the way, are related to the ones in legumes. So like if you plant alfalfa or beans or something like that to fertilize your soil, those plants have associations with bacteria very similar to these. So those plants and the algae that live inside of corals are similar in that way. So corals, especially hermatypic corals, will actually take in these bacteria and then use them in the same way that they do zooxanthellae, but they have a different role. They're nitrogen fixers instead of carbon fixers. So they supply nitrogen to the zooxanthellae. So you can actually say they benefit the algae in the coral more than the coral itself. It's an indirect association.
Gotcha.
People say, hey, they say it's a coral holobiont, right? So they say it's like a super organism with all kinds of bacteria and stuff. But bacteria such as this play a huge role because they provide that nitrogen. But again, so so you're asking do you use it every day, right? Well, you wouldn't necessarily have to, because these bacteria would grow quite well in an appropriate environment. If we perfectly mimicked the reef environment, they would be there all the time, and they would actually be, one study found them to be among the top 18 genera, Rhodopseudomonas, on a healthy coral reef in the Great Barrier Reef, out of hundreds or even thousands of genera, right, potentially, that are on a coral reef. So extremely abundant in those environments. But if you have a polluted like an aquarium or an actually polluted reef, they found off on a Hawaiian reef even, where there was a lot of sewage or some kind of waste going into the water, increased the nitrate. These bacteria occurred at a much lower density because they weren't as competitive. It's that ability to create nitrogen that other bacteria don't have, the nitrogen fixing, right, where they make their own ammonia out of nitrogen gas. That's a huge advantage in a nitrogen limited environment against other bacteria. So anyway, that's why they don't grow in an aquarium well normally, unless we actually mimic a coral reef environment and have zero nitrates, essentially. But you can still get the benefits from dosing them like this. Sure. So if you do it every single day, yes, that's better than doing it every other day or once a week. That's not to say that doing it once a week doesn't have its benefits. It's just that to enjoy the maximum benefits, you could feed every day. And it seems, I guess the easiest way to do that would just be to do that when you do your actual feeding with foods and everything, because you would just mix it in with everything. Most people have I don't know, you probably have like a cocktail of frozen foods and right?
And I soak it with vitamins and all that stuff. And so I guess that leads into another question. So I'll usually do my my batch of food will probably last me 2 days, typically, maybe 3. I try not to make more than 3 days because I don't want it going bad in the fridge or anything. But then I'll throw in all kinds of vitamins, Celcon, and all kinds of other stuff. If you were to put your, throw on some of that bacteria, would it be good in the fridge for say 2-3 days mixed in with the food, or would that kind of foul the food after the initial one?
Yeah, it would never foul the food because it doesn't produce any harmful metabolites. Like the refrigerant Pseudomonas is the one that spoils food in your fridge and produces harmful, the death smell that we're familiar with. These bacteria don't really do anything like that, but they don't really grow below like 75 degrees anyway. They like it very warm. So they would essentially be dormant at a refrigerated temperature anyway.
Okay. Yeah. Cool.
Technically, even at room temperature, they would just kind of flatline. And in so doing, they might even they might even protect your food from spoilage, because they would compete with other bacteria that might actually be more harmful.
Okay.
Yeah.
Very cool.
So, yeah. And for anyone interested in looking into that more, like the particular nutritional benefits or anything like that, like I said, you just do a Google search for like purple non-sulfur bacteria and probiotic and maybe aquaculture or something like that. You'll find all kinds of different ways that they put it. So like if you use pellets or something, I'm sure out there somewhere there's somebody applying it to trout chow or something like that, right? They give you like a specific time to soak it and all that, and you can kind of model it after what they did. But people with reef tanks, I never even really recommend using the binders, because when we started out, we had people just throwing it into the tank anyway. And then that we'd expect the fish to eat it vicariously, more if the like fish grazing algae that had it on it, or copepods that had eaten it and things like that. And then we realized more later that it was more advantageous to put it in the foods and get the most use out of it in their gut. But even then, a lot of it just comes off, like I said. Sure. So that's the easiest way to add it every day, though, right? Is if you just put it in their foods.
Yeah, exactly.
Yeah. And then for anybody out there that actually, if every community's got a clownfish breeder or something like that, and right, maybe not someone watching it right now, but later, there are lots of there's a good amount of literature out there where people actually gut load live food. So if you take it one step further and you feed it to your live foods before you add those to the tank, they definitely do eat this stuff, and those nutritional qualities and probiotic qualities definitely are conferred to the cultured organism, be it a fish or shrimp or whatever. So definitely really useful. So I was asked if there are any other questions. I could ramble on forever. You probably can already tell that, but
yeah, I was going to say, so that was the probio bacteria that you offer, right? The yellow bottle.
Yeah,
because you have 3 different types.
Oh, I lost you for. Oh, now I can hear you again. That's PNS. PNS ProBio. So the red just kind of indicates it's the live. They call it purple non-sulfur, but these particular ones are more reddish orange. So we have the red cap. There are other species, and then they'll end up having different colors, but those are the live ones. And then the yellow cap that you saw is PNS yellow snow. That's made with these bacteria, but that's actually a prepared product. So that's not live. It's fermented with these bacteria, but then it's pasteurized midway through the process, because it simulates a marine snowfall. So it's basically like a fake marine snow. But maybe the closest thing to the real thing that there could be, because it's actually a microbial process that forms it, but it is it's not a live product. So you'll get the nutrition plus some other stuff that's in there as part of making that marine snow simulation. But it won't have the probiotic qualities, or it won't be able to perform denitrification and so forth. It's almost a supplement.
Okay.
That
Gotcha.
might be worth mentioning actually too, because a lot of people sell them together, and that's for good reason. Yellow snow is actually a companion product. So these bacteria are pretty actually regarded by ecologists as one of the most versatile organisms on Earth, because they're, anaerobic or aerobic, saltwater, freshwater. They can perform nitrogen fixation and denitrification, complete opposite ends of the nitrogen cycle, and just all these extreme environments. But one of their Achilles heel, I guess, each one of them has a different B vitamin need. So those are called cofactors if you're culturing these bacteria. And anyway, yellow snow yellow snow has the entire B vitamin complex. So could actually, when you use yellow snow, you're actually promoting the growth of the bacteria we sell, because it has what they need to grow as well.
Gotcha.
Yeah. It's particularly rich in B12, which I mentioned earlier. Only none of our bacteria have a requirement for B12, but it does help them grow. But B12 is important even for even zooxanthellae have a B12 requirement. So it kind of feeds the whole reef tank, from the bottom up. Copepods eat it, corals eat it, any kind of filter feeder. But even the bacteria can eat it. And that's kind of why it was developed as a companion product to the bacteria we sell. So it kind of completes the base of the ecosystem nutritionally, and also in terms of particle size. So the live bacteria are only like 1 or 2 microns in length. The huge ones maybe 3, but some of the particles in marine snow, of course, are large enough to see with the naked eye. So it kind of provides feeding opportunities for a much broader range of filter feeders. So a lot of feather worms and certain types of sea cucumbers and stuff that might prefer larger particles. I'm only guessing. I don't know what size particle they eat, but different things prefer different size particles, and it just kind of broadens the range, what we can feed this stuff to.
Okay.
Yeah. And then the third product is called substrate sauce, and that has more of like a fuchsia kind of purpley cap. Not as many people have seen that. That's not as widely used because it's strictly a cycling product. Not too much different about that one. It has Rhodopseudomonas in it, the same purple non-sulfur bacterium that is in our other products, but it has an additional purple non-sulfur bacterium called Rhodospirillum. I wouldn't necessarily expect you guys to remember all that, but has an additional spiral shaped purple non-sulfur bacterium in it that has that kind of fuchsia color to it. And the only reason that's in there is because we developed that product after we started growing that other bacteria. No other reason. What makes that a cycling product is how it's grown. So we put ammonia in there as part of their culturing, as part of the culture medium. So the presence of ammonia stops nitrogen fixation, meaning they won't make their own ammonia if they have it in the environment, which is what you'd want them to do in a newly cycling environment. And that was also cultivated in that's also grown in saltwater. It's the only product we have that's specifically saltwater. And the reason for that is, like, if you're cycling your tank, you want them to start growing in your tank really fast. So that shortens what's called the lag time, where the bacteria are just kind of sitting there adjusting to the new environment before they go into exponential growth phase. So yeah, salt water is the other thing, and that has a slight excess of phosphorus in it, phosphate. So a cycling tank, especially nowadays, where people just use dry rock and they do fishless cycling techniques, most of the bacteria starve for phosphate, and it limits how fast your cycle can occur. We actually, full dose of substrate sauce raises your phosphate by approximately 0.05 parts per million.
Okay.
Yeah. So then the main thing of that is because they're denitrifiers, you would expect after the cycling process is over to start, instead of just seeing your nitrate accumulate and then you go, my cycle is over, you go through the actual full cycle then, where you start to see the nitrate go down, assuming only one thing, and that's that you've provided a suitable anaerobic habitat for them. So like some marine pure or some kind of porous biomedium. And the last thing I'd say about the products is that in the somewhat near future, we're going to make a change where we basically combine the two. So ProBio will just will take on rhodospirillum, which is the second one that was in substrate sauce, and the Rhodobacter, a third purple non-sulfur bacteria that we grow. And so it'll have 3 species instead of the one, and they just kind of complement each other. And then we'll just have that one product to make things easier for people, because I totally understand if like this isn't what you do, like all these big weird words and
you don't know what to choose, right?
yeah, well, and we don't, a lot of people don't know in our hobby much about these bacteria. We're stuck on the nitrifying bacteria that actually ecologically are not that important on a coral reef. They play a pretty minor role. That's not to say they're not valuable to us as aquarists, because we're starting out with just like what would be like a disturbed environment. So we have to go through that ecological succession. But after that's all said and done, you start adding fish and everything. Heterotrophs rule that world, and heterotrophic bacteria. So you don't really see a lot of nitrification going on. So, one nice thing about having these guys in there from the beginning is that you have the capacity to degrade organic matter, which nitrifying bacteria do not have. You have the capacity for denitrification, which nitrifying bacteria do not have. And these bacteria, even though they're anaerobic and they live in like biofilms eventually, if they survive in your tank, the daughter cells are motile. So they swim around and they swim around in the plankton. So even when you're not actively dosing the product and feeding, if you have a resident population of these bacteria in the water, they're constantly sending out these daughter cells that go out swimming into the water column where they can get consumed by corals, tridacnid clams, stuff like that. So in that respect, they're kind of like the gift that keeps on giving, I guess. You just put a little bit of, put a little TLC into cultivating them, just a little bit of biomedia, right? Because no one wants deep sand beds anymore.
Right. Do you see any issues using UV with the bacteria, just in general, I guess? I know there's a lot of mixed feelings out there about doing that. What are your thoughts on that?
I think it's bad. If you're trying to replicate, the natural marine environment. So I haven't seen a study showing that these bacteria are killed by UV, but I can only assume that they are. All little tiny organisms, especially bacteria, are killed by ultraviolet radiation. For those few of us that actually maintain our UV sterilizers properly, right? Where they're not, the flow isn't too high, or they're underpowered, or the bulb is 5 years old, or the sheath is covered in film or precipitated whatnot. But yeah, if they're operating like they're meant to, they're killing these bacteria, and they're also killing some of the most important ones in any coral reef, like the Pelagibacter group. I don't know if you guys are familiar with Aquabiomics.
I've heard about it, but I haven't really done anything with it.
Okay, so Dr. Meyer's the one who runs that. He's a coral biologist who got interested in microbiology, and that's how he ended up starting that company. But he does a lot of case studies with the samples that people send him, and he's found that people that use ultraviolet sterilizers have much lower abundance of this really important group called the Pelagibacteriaceae or something like that. The Pelagibacter is the main genus, and they're like they are the most abundant organism on Earth, certainly in the ocean, and definitely the most abundant plankton. In terms of numbers and probably biomass, they're just really abundant and really important to corals. Corals eat them, and they also consume some byproducts that the corals produce. DMSP, for example, which is gives the ocean that ocean smell, that sulfury kind of. So they consume that sulfurous compound, which is important ecologically. Anyway, yeah, if you use ultraviolet radiation, we've already noted in the aquarium that this particular really important group, that's one of the most important coral foods, to the extent that corals produce mucus in their substances in their mucus to attract them in order to feed on them. That particular group of bacteria is wiped out by ultraviolet sterilization because they're completely pelagic. They only live in the water column, so they're totally susceptible to getting pulled in by a UV sterilizer and killed that way. We recommend for anybody that is trying to seed their tank with our bacteria. A lot of people make an attempt to get it to grow in their tanks. Sometimes some people are successful. We've done aquabiome tests of one of the reef builder systems. Jake was interested in these bacteria before he passed. He was using it in the studio, and a little bit after his passing, I was able to get into the studio and take some tests. And Rhodosumonas was present in those tests. So, and he famously hated deep sand beds and stuff like that. Right. So these bacteria found places in that environment to live, which was very simple and very plain. Not just from what he says, but if you've ever been in the studio or saw the systems, they were really clean and simple. Not a lot of anaerobic microhabitat.
A lot of bare bottoms and minimal rock work and stuff.
Totally, yeah. Lots of corals, though. And maybe that's where they lived. So that'd be a good place to jump in with the association between corals more if you guys think that'd be a...
Sure.
Yeah. And by the way, just cut me down at any point if you have a question, but there's kind of a normal line of thought, I guess, with how these work from the bottom of the ecosystem up in a reef tank. But one of the big things then, I guess, is corals, because that's the corals are the foundation of the reef. And that's probably the most complex thing that they do, I guess, is their interaction in the coral holobiont. So as I mentioned, corals will actually farm these bacteria. Rhodosumonas, for example, palustris, the specific species that we cultivate, has been found in corals in all kinds of places. Caribbean, Pacific corals, just kind of a ubiquitous feature of the coral microbiome. And as I noted, and I know I lose people at the nitrogen fixation thing. That's a new concept to a lot of aquarists, but it's actually a really important one if you're trying to mimic the coral reef or even just understand the coral reef. Because again, like now I mentioned the Darwin's paradox, where it's people can never understand why corals could survive in such a nitrogen limited environment, right? Well, part of that is corals don't compete well with algae. So you go to like the Pacific Northwest or some of these environments that outside of the tropics where there's more upwelling and they're much more nutrient rich. Corals don't grow well in those environments. It's certainly not like hermatypic corals like we have, the photosynthetic corals. And that's why? Well, mainly because those are kelp beds or, seaweed beds. They dominate over corals. They're just faster growing, and they have other ways to compete with corals by poisoning them essentially, or just smothering them. So in a coral reef, it's a little different because it's a nitrogen limited environment. It limits of course you have your herbivores and they play a role too in controlling algae, but the main influence there in that interaction between corals and algae is the nitrogen limitation. So what happens is that the water column, if you ever go to a coral reef and you test nitrate, nitrate's usually like I saw one like 0.006 parts per million nitrate. It's like a normal. They say so-called pristine reefs can even be less than that. They're essentially zero nitrate. And of course, any ammonia that's produced is it's not gobbled up by nitrovirus and converted to nitrate. It's pretty much just assimilated into bacteria and other things, corals too, there, and just used directly. But it's in very short supply. So because the water column doesn't have a lot of nitrate in it, you don't have a lot of algae. If you've been to a healthy coral reef, that's a conspicuous difference from other environments you may see that are just covered in seaweed. Well, so then how are corals so productive? And that's the paradox, right? Well, it's because of these bacteria that live on and in them. And different types of, so you have really just a couple groups of diazotrophs on a reef. You have obviously the cyanobacteria. They are important diazotrophs in a lot of environments. On a coral, they tend to live in the mucus on the outside of the coral. And then you have, it's like an archaea or something like that, kind of a more obscure one. And then the third really big important diazotroph is these purple non-sulfur bacteria. And they are really well represented in the coral microbiome because, they have all these beneficial, for one, they compete with Vibrio quite viciously because they produce streptomycin, these other antibiotics that naturally inhibit the growth of Vibrio. So they control how much Vibrio is. There's always going to be Vibrio growing on it, even on a healthy, normal coral reef, but they suppress that somewhat. And they live not in the mucus like some of the other diazotrophs, like your cyanobacteria. They live inside of the coral, in its gut. And so they are there supplying one, they call them leaky. They say that they leak a little bit of ammonia when they're so they're fixing nitrogen. They're making ammonia mainly for themselves to grow because there's no nitrate or ammonia in the environment, or very little of it. So when ammonia, so we'll say when nitrogen, biologically available nitrogen, is lacking, then they switch to nitrogen fixing mode. They start making their own ammonia and they start growing. Well, they say, one study I saw, they figured a coral reef is about the nitrogen on a reef. We're talking like the tissues of the corals and clams and fish even. Ultimately, it's they call it diazotrophically, I can't remember, DIN. But basically, it originated from a diazotroph, right? That nitrogen, it's about 50%. So you could probably safely say 50% of a coral's nitrogen doesn't come from the plankton they eat, but specifically from those bacteria that live inside of them.
Okay.
Right. So the bacteria will leak the nitrogen, and then the zooxanthellae can take that directly. And then the other thing is that the corals actually eat these bacteria. So they kind of farm them like corals also eat zooxanthellae.
Right.
If they have excess, they'll just consume them. They have ways of regulating the content that they're farming, so to speak. And they do the same with these bacteria, and they can take them in at different rates too. So in one instance, there was a study they found where there was a seasonal variation in the amount of ammonia and nitrate in the water. They found that corals took in, it was specifically Rhodosudomonas palustris, the bacteria that's in probio. It actually varied. So at the times of year when nitrogen was even less available than normal, there was more Rhodosudomonas in the coral. So they kind of regulate. There may be that there are more Rhodosudomonas in the water when nitrogen is low, because they're better able to compete at that time, right? Because they have that ability to make their own nitrogen anyway. So it could be zero ammonia, zero nitrate. They'll still grow because there's plenty of nitrogen gas in the water. The atmosphere is 80% nitrogen gas, so they're never going to run out of, as long as they can turn it into ammonia, they're never going to starve for ammonia, right? They found that when there was seasonally low availability of nitrogen, the corals actually took in more of these bacteria and maybe consume them less and allowed them to do their thing, which I thought was pretty cool. that, these associations are so ancient and complex between the corals and these particular bacteria that makes you wonder, like, how often we import these bacteria into our tanks. When we buy a coral, a wild one, or even maybe a captive bred one, even, how much this is getting passed around and we're getting it in the tank. It's hard to say, but the corals do lose. We always have this idea that, we keep adding things to our tank and we're increasing the biodiversity of the bacteria, because we just assume every time we add something, it's just a net gain. But that's not really how it works. Things die off too. We have things dominate in an aquarium. And then, especially in an aquarium, a small closed environment like that, and not a particularly dynamic one either compared to the wild, where you always have recruitment from outside, habitats. What tends to happen is biodiversity, microbiological biodiversity tends to decrease over time. And we've seen in corals, even in the wild, as a coral colony gets older, it's a symbiotic bacteria. The bacteria that live on and in it actually become less diverse over time. So, though we haven't looked at this in captive corals yet, my guess is that over time, and this will get more important as we rely less and less on maricultured corals and more on aquacultured corals, that we're going to start losing some of the natural biodiversity that we take for granted, or we once took for granted when we buy these corals. That over time, they'll get less and less, and then they'll become less resilient to different types of diseases and nutritional deficiencies, things like that. So, in a way, because these bacteria are so abundant in the wild, but not particularly abundant in aquariums, it's just kind of a way of rebalancing. It's like I like to call it a reset button for your microbiome. So you can kind of increase this naturally abundant group, restore it to some abundant state. And, they may get consumed by corals, and then on a slower scale get outcompeted by other bacteria, but you can always re-add it and kind of level the playing field again.
I'm so glad that you make these products and make them available for us so that we can reset and we can improve our aquarium. You have brought so many very interesting, to me, it's some new ideas that I'd never considered. This has just been so fantastic. Does anyone have any, because we're at time, does anyone have any burning questions for Ken? Because Ken, we so appreciate you coming out and sharing your evening with us. And he looks fantastic. You guys probably don't know, he's also the father of a very active 6-month-old, and he has other preparations he needs to get back to. And so I want to be respectful of your time. But I, on behalf of the club, we are so honored that you came out and shared everything with us this evening. Thank you so much. Nobody spoke up with questions, and since we're over on time, I'm going to do something really fun. It is Valentine's Eve, and so to give love back, instead of the usual drawing, both Nick and Ron, I have 2 tote bags. And these are donations from Odyssey Pets in Dallas. And inside each tote, now they may be slightly different, but they've got some products to help you take care of your mixed reef. And there's some stickers and there's some food. And so I'll get with you and find out if you want me to mail them to you, if you'd want to pick them up at the frag swap, whatever works for you is for us.
Just bring mine to the frag swap, Lisa.
Yeah, absolutely. Yeah, no need to mail it.
Okay. Oh, and Ron, when you speak, we get to see your reef, and it looks spectacular.
Oh yeah.
Yes. Thank you. I was
Oh, you're welcome.
Thank you.
Here you go.
Nice.
Oh, looks great.
Very nice.
Lots of money into that thing.
That's how it goes.
I used a lot of phyto. I tried to make a culture. I keep killing my cultures, so this product that you're talking about, I may have to check that out. As an alternative.
I don't know if you'd have better luck with our stuff, but we do have a grow your own kit that is innovative. That's the only thing we sell direct to the public.
Oh yeah.
Yeah. So we have a distributor now. We had to we pulled our store, so it's hard to find, though. It's just hidden. If you you can just find it if you go to Google and do PNS home grow. So home grow is one word, home and then GRO. And that makes looks like you've got a pretty big tank there. So it's a economic way of using these bacteria. It's basically the same thing as growing phyto, really, except it's anaerobic, so you don't have the bubbler.
Yeah.
You just put them on light and keep them warm. And 30 days, you have about 8,000 gallons. You can treat enough to treat 8,000 gallons.
I was going to ask you about culturing, but I didn't want to step on your business there. But so I would definitely look into that because I'm always into that kind of stuff. As you can see, I have a lot of coral, so I have to go through a lot of micro foods.
Yeah. And these are, it's like a live food, and you have it on, right on hand like that. So you don't have to worry about refrigerating it and having it go bad or anything like that. And you can produce one big batch. So that if you make like that one cube, they retail for $140, by the way.
That's not bad.
Yeah, you do the math. It's like treats 8,000 gallons and doesn't produce. It has the additional benefits of. Again, it doesn't. A lot of the economics that go into this are kind of like, okay, other foods are actually going to pollute the water, so I'm going to have to use more chemical filtration. You do more water changes, meaning more RO water. My filtration system, the cartridges get used up faster and stuff like that. So it's you factor all that in, it's actually pretty economical, but the kit's even more so because it's like, that's an enormous amount. And as long as you keep it in that cube on the light, they're viable for like a year.
Oh, wow.
Yeah. The cool thing is that, because they're photosynthetic and they produce all those pigments, you can see that they're there. So that the bacteria is there. It's a characteristic like reddish purple color that they turn. That kit, by the way, has all 3 species that I mentioned in it. So
what was that? What do I search again?
Yeah, it's just a PNS space home grow.
Okay, PNS space home grow. Okay, perfect.
Yeah, and the home grow is just a HOME GRO, all
GRO. Okay,
yeah.
Gotcha.
Yeah, and then it'll take you to the right to the listing. You should see like 2.5 gallons on there.
Okay.
How much it makes at the top of Google. And so those, like I said, those last a really long time too. So even if you don't have a humongous tank, that's something you can, kind of comes with a spigot and a collapsible cube. So you don't constantly, some people will fill up a big bottle and then use that periodically, but you can just keep, you almost have it like on tap.
Yeah, that's nice.
A live plankton on tap that you just crank and feed what you want. You can't overdose it. And,
it's kind of what I do with my phyto. I have it in like those big, I guess they're picnic tea things. They're like 1.5 gallons, and they have little spigots. So I just pour it out and then add salt water and fertilizer. What
species are you growing?
I just grow nano, and I just killed it last week. I don't know what happened, but when the light came on, my jar was clear. So I don't know. Something was in there, ate it all. But some other bacteria was in there or something. Who knows? That's the hardest part, is keeping all that sterile. Just keeping it all clean.
Yeah, it's tough.
That seems to be the hardest part.
Totally what we learned, like where I was growing phyto on a commercial scale, is you can't just inoculate a big swimming pool, with a bottle or something. It's something else will take over and kill it all. You have to keep adding incrementally
Yeah.
larger, in a little test tube up to a jar, up to like a tank thing, before you put that in the. So there's always contamination and stuff, but if you keep up with the phyto, or even if you just buy the phyto, like I said, if you use these bacteria and the phyto together, you have compounded nutritional benefits because they actually are nutritionally complementary. And by the way, if you use any other probiotics, I don't know of any other products in our market that use these, but they use a lot of lactic acid bacteria like lactobacillus and Benepets, for example, stuff like that. Well, these bacteria are found to promote the growth of those bacteria. So they actually they don't really compete with each other or outdo each other, or they're not even redundant. They actually potentiate each other. So you get more out of each one using them together. So that's kind of, yeah. So yeah, again, a lot of stuff. I should probably end with this. If there's a lot of other information, it's like, and Nick, man, thanks. I bombarded you with information. I had like one face to look at and I just went for it, but you're a great sport. But if you guys are like, or anyone out there is truly interested in any of this stuff and wants to dig deeper, our website is, it's not .com, it's hydrospace.store. And we have a blog there, and I contribute to it, but we also have Taras Pleskin writes a lot. He's more of a phycologist guy, a microalgae guy, but he writes for Reef Builders, and he's really smart, and he's written some great.
Right?
What's that? Yes, exactly. Yeah, great guy all around. So he writes for us as well. We have a lot of really informative and in-depth articles in the blog, and most of them they either have a list of references or they have links in the text to direct you to the peer-reviewed literature that we reference in those articles. So most of the claims we make, it's not witchcraft. We're usually referring to some very specific study that demonstrated these bacteria do a very specific thing or under a certain circumstance. And anyway, so it's a good place to get additional information. So I'd invite anybody to go check out our blog at hydrospace.store.
Cool.
Awesome. Thank you so much.
Yeah, thank you. This was wonderful.
Appreciate the time.
Thank you. Thank you very much.
Hi, everyone. It's Ken from Hydrospace. Looking a little disheveled. It is a Saturday morning, but we just got a request for a video tour of the facility. I guess some of you are interested in seeing the place after our interview last week, which we appreciate. So just thought we'd bring you through and show you everything we've got. Heads up, we just did a massive bottling. So the place is about as disheveled as I am right now. But at least you'll get an honest and authentic view of what the place looks like after the end of a long work week. So here we go, just starting out in the office here. For starters, if you're wondering why the reflective material everywhere, we maintain a temperature of just about 80 degrees Fahrenheit down here, 78 to 79 generally. Just to kind of keep the ambient air temperature up. That's because we don't have individual heaters in all of our culture vessels. That would be these 5-gallon collapsible containers. I was mentioning that we just did a big bottling. We have really low inventory right now, so that's why there's so much stuff missing off of this shelf. We did a bunch of Rhodopseudomonas, which is the product that is in PNS ProBio, the one bacteria, I should say. That's what the color of this guy is. So this is the reddish orange pigment that we referred to. A lot of those carotenoids. If you stir it up, it turns almost like a rich brick red. But this is the centrifuge we just got. You guys are the first to see it. 1.5 horsepower, so it's a pretty powerful guy. We're gonna make paste out of that, and with the paste, we have some additional equipment for drying. That's going to be a more proprietary process, but with those powders, we have to test to see if they'll be viable, and if so, we'll be able to include those in some other products, both for aquaculture and agriculture, the latter being more to our interest right now. What you're seeing on the floor are completed culture vessels, so they're full of growth media, and they're ready to inoculate and get up on the shelves under the light. Lights are just regular old 5,000 lumen shop lights, so nothing anyone wouldn't have just sitting around at home. These guys aren't particularly finicky about the type of lighting that they can use. They're more particular about warmth, so that's why it's a blistering 78, 79 to 80 degrees down here. Makes it a little rough to work. Usually I do half my work in summer clothes down here. These racks are for other species. So, as you know, we do have, for example, a Rhodospirillum, which is more of that nice purpley red color. That one is the second species that is in PNS substrate sauce, our cycling product. And then Rhodobacter is more orangey. It even starts out almost greenish, goes through a lot of different color phases before it ends up kind of a nice rich red. But these are all different. Like Rhodospirillum spends more time in the water column, so it is a better as a coral food, whereas Rhodobacter likes to live more in biofilms. So that's a better one to grow on substrates, say, to inhibit the growth of cyanobacteria. But they're all purple non-sulfur bacteria. That's kind of what that's what we've specialized in. So again, anaerobic, that's why you don't see bubblers all over, like if this was like an algae. We seal them up and actually keep them anaerobic throughout the entire culture process. So it's also nice and quiet down here, with the exception of the fans, many fans and forced air heaters we have running at the time. Hard on electricity too, believe it or not. With I don't know, 24 solar panels, we still have electrical bills of $600, maybe $700 on some months. But it's all these heaters sitting all over the place, blowing air around all the time. It gets pricey with the addition of these lights. We blew out our power many times before we finally had someone come in and put in this sub panel, so this controls everything down here individually. And luckily, we have a battery power now on our solar, so if something happens here in Colorado, we get a heavy snowfall, something like that, and the power lines go out, which often happens with the wet snow, trees go down, take out power lines. We're out in the country, we're the last to get restored. Sometimes it's good to have that backup power and keep everything going down here. More the heat than the lights, to be honest. So coming around the corner here, just a lot of supplies and packaging, boring stuff. But here's where most of the action happens. This is what we call our kitchen, the kitchen. So we got these big 7-gallon, well, not really big. It's actually in term scale of processing, it's kind of you'd say it's almost craft or something. It's a pretty small batch, really, the way we brew things, but a couple of these 7-gallon kettles at a time, so really a couple cubes at a time we brew. So that's nice for quality control, to be honest. Even if we upscale, we'll probably just multiply these small batches rather than making bigger batches, and that way we can keep track of contaminations and things like that. But a lot of these surfaces double for things like, for example, we do a lot of our microscopy here on this screen, but when it's time to put labels on, this same table just serves a different purpose. This is our future CEO's office. But anyway, yeah, so spend more of my time here than anything. I like to joke. That's the sink, of course, and yeah, a lot of washing.
a lot of washing. So not always exciting down here, but nevertheless fun, just getting to discover new ways to grow this stuff and work with y'all. So hope this helps give in addition to a face, like a place to kind of connect with these products and give you an idea how they're grown in addition to just what they do or what our label looks like and so forth. So thanks again for checking out the interview the other day. I understand they're making a video, so I did promise to answer any questions. So if I find questions directed to us from anywhere, whether on Instagram or YouTube or anywhere, if I can find it, I will answer it. So hopefully you got some of those coming in, and I'll hear from you from there and other places. So thanks again. Take care. Have a great weekend.